Hello everybody, so we're about to start the Flash Talk session. Brief reminder, there's going to be 3 minutes per talk. There's no times for questions. And for all the speakers, we're going to put a 30 seconds reminder, where's your 2 minutes, 30 seconds. So now we have Saskia Stenzel, which is going to talk to us about cell-to-cell spread of SARS-CoV-2 rather than transmission, rather than cell-free infection.
So my name is Saskia. I'm working as a PhD student at the Goffini lab at LSTM in Liverpool. And my current research focuses on SARS-CoV-2 cell-to-cell transmission. So in general, it is known that envelope viruses spread via 2 transmission routes in cultured cells or tissues, either via cell-free particles or through tight cell-to-cell context. For the cell-free infection condition, newly synthesized viral particles assemble at the membrane of a producer cell and are then released into the cytosol. With the attachment to a cellular receptor, the virus is then able to enter and further to infect the cell. The latter mode of transmission usually involves tight cell-to-cell contact, leading to an increased local viral particle density and to an efficient transfer of virus from cell to cell. But why is it actually interesting for us to know how virus is being transmitted within tissue? For HIV-1, it is already known that cell-to-cell transmission is much more efficient than the infection via cell-free particles, and furthermore, it allows to escape from neutralization by monoclonal antibodies, which are directed to viral glycoproteins. In order to investigate this in the context of SARS-CoV-2 infection, we would initially infect cells with the recombinant SARS-CoV-2 GFP reporter virus to collect the virus containing supernatant after 24 hours to treat cell-free naive cells for the cell-free infection condition, and we would harvest the donor cells to set up a co-culture also with naive target cells for the cell-to-cell transmission condition. Upon further 48 hours, we would harvest samples to check for GFP positivity, indicating de novo infection using flow cytometry. Under these experimental conditions, we typically observed similar infection efficiencies for both transmission modes using a Vero6 Calo3 co-culture setup. This was actually quite surprising, as we detected a clearly higher abundance of viral proteins in the cell lysates of donor cells compared to the virus-containing supernatant, indicating an increased viral titer being directly transmitted from cell to cell. This is why we hypothesized that one of the modes may act earlier than the other, and being therefore maybe more efficient. And indeed, after performing viral replication kinetics, harvesting samples at specific time points, we detected a clearly higher percentage of GFP-positive cells in the context of direct cell-to-cell transmission compared to cell-free infection, indicating that cell-to-cell transmission is actually working earlier. Continuing by investigating the functional relevance of the human ACE2 receptor and the serine protease TMPRSS2 by using specific inhibitory agents, we could see that both transmission modes are dependent on the ACE2 receptor, whereas the TMPRSS2-mediated cleavage of the spike seems to be just beneficial in the context of cell-free infection. But this was not the only difference we found. Using several monoclonal antibodies which are directed to the spike receptor binding domain to pre-treat either virus-containing supernatant or donor cells revealed that SARS-CoV-2 cell-to-cell transmission exhibited an increased resistance to antibody-mediated neutralization of the spike, whereas cell-free infection was quite sensitive. And this was further validated by characterizing 2 antibodies which showed particularly strong inhibition capacities compared to isotype match control, which was non-reactive in titration assays. But for further details and future outlook, how we want to investigate the mechanism in the future, I would be happy to welcome you at my poster 3. And thanks for listening.